few things I'd like to discuss with you. Before going to that, as we discussed offline and online also about the anatomical features about the stem anatomy and all, a brief some slides I would like to share with you. Then we'll proceed to the recent trends in taxonomy. Just wait for a moment. I hope you are able to see the screen. So this is, are you able to see the screen?
Yes, ma'am.
Yeah. Sorry for the delay. So this is what we had discussed about the Boerhaavia stem. As the Boerhaavia is a herbaceous plant, in fact, but it shows secondary growth, which is not the usual phenomenon. So it shows abnormal secondary growth, which you are going to see the things in practicals as well. An important material for the section cutting in practicals as well. So here basically the abnormal anomalous secondary growth it is called, as it is not the normal growth. Okay, so these are the medullary vascular bundles which are half moon shape. So by observing these two vascular bundles itself, you can identify the stem when it is given. So these are the medullary vascular bundles. Normally in a dicot plant, the vascular bundles are at the peripheral, right? So they just a ring of vascular bundles will be present at the periphery, and it forms a continuous ring like at the beginning of the secondary growth. But here from the beginning there will be two vascular bundles in the medulla surrounded by another ring. So here the cambium which is present in each vascular bundle uh, functional for a short period. It is not going to make a ring-like thing. Just a brief amount of the secondary growth has been taken place in these two vascular bundles are again here, and there is another cortical vascular bundle is going to form that is going to add the secondary tissues. So here there are the medullary bundles, primary medullary bundles, and the secondary bundles formed in the cortex region, which will continue for a while the secondary growth. So here the abnormal behavior, also abnormal arrangement of the vascular bundles in the primary structure, abnormal formation of the ring also been seen. So this is the thing which you are going to study in theory, which is important, plus in practicals also this is one material you are going to see. Okay, so this is other form of this is begonia is another example which I discussed also in the class. So here the normal secondary growth happens with the activity of the cambial ring. So the cambial ring by dividing the cells, one row of the cells remain always meristematic, and the other cells which are generated will get differentiated into the cells facing inside will be differentiated into xylem, that is called secondary xylem, and the cells facing the outward are going to get differentiated as phloem. So here, as interestingly as you can see, at 4 places the cambium, instead of developing the xylem elements inside, develops the parenchymatous tissue. So at four corners we observe this second, the instead of the xylem, it is going to produce the phloem tissues inside. So four at cross four phloem, intruded phloem is seen in the secondary xylem. That is one peculiar feature we observe in the begonia stem, bignonia stem. So this is another form of anomalous secondary growth. So this is actually with the cambial ring behavior in a abnormal way. This is what actually just now I told. Instead of developing the secondary xylem inside and secondary phloem outside, at different places it develops only the parenchyma tissue. So there is a huge area of medullary, normally thin rows of cells will be seen called as medullary rays in the secondary xylem, but here a very broad area of the parenchymatous tissue is going to form, as labeled as conjunctive tissue, that is simply parenchyma. The secondary xylem, normally it will be a continuous thing and forms a compact ring-like structure. But in the case of this Aristolochia, it is a cleaved secondary xylem cylinder, which is very unique, and it is not going to make any hardwood also because of this reason. Instead of developing the xylem inside, it is going to develop a continuous parenchyma tissue in a broad area, making it is like a cleaved or separated, not as a single cylinder. So this is one form of abnormal behavior of the cambium ring. Aristolochia is also an example we have seen. So this is what actually you can see the overall view, how it will be reflected as. Okay, so this is how the deeply separated xylem cylinder is being seen. And these are some of the abnormal behavior only. The growth, here Tinonia and Sarjania, these are the different examples. It is there in our book as well. So this is a Bahinia, is actually a tree member. Bahinia is a Salpinaceae member. Devakanchana it is called in Telugu. But in some of the members of the Bahinia Valley and also Bahinia languistiforum, here the growth happens in a very abnormal way. So it is not normally a stem will be of round in structure in the circumference wise, but here it is the growth happens very smaller at one point and much more in the side region. So it becomes a flat structure. Particularly this kind of abnormal behavior of the cambial ring, activity wise, it generates more tissue on one side and very less tissue on another side, makes it possible the stem to grow as a liana. Liana is a woody climber, and these are different vascular bundles are going to be present in another form of abnormality. like Thinonia and Sarjania, where each cylinder is separate. So the secondary growth happens separately in each cylinder. So this is another example. And this is the gymnosperm plant, the neatum, which I told you very much similar to that of angiosperms. Netials members are very close to angiosperms in several features, and because of that we consider the Netials group with the Netum and Ephedra, Welwitschia examples we have studied. It is more advanced among all the other gymnosperms, right? So here this is one example, Netum vula. This is also a liana. Normally we observe concentric rings, is it not? Just a circular form of rings one after the other we'll see in the normal secondary growth. But here the abnormal behavior, here behavior means here nothing differently, but here the growth happens one side very less and other side very high. So that makes the stem can grow around, on another support, being here this netum plant also, and this netum vula also a liana, a woody climber. This is more abundant in the Araku region, also in the Nalamala forest. It's a very unique plant, netum vula, and also endemic in a way. So this is another example of Nyctanthus. Nyctanthus is Parijatam. So we are all known that fragrant flowers where the stalk is orange in color. Some of you must have seen this plant Nyctanthus. So this also has a basic different anomalous arrangement in the beginning itself. So they have, normally this is the form of vascular bundle arrangement, right? So this will show the this ring of normal vascular bundle ring shows the normal activity only of the secondary growth, but there are at 4 angles actually the Nyctanthus will have an angular stem. So in the angles we observe these are called as cortical bundles. So the cortical vascular bundles which are in the inverted position. So normally we know that the xylem is an endark condition. That is a standard feature, as all of you can clearly see. So these are the metaxylem points towards periphery and the protoxylem points towards the center, right? So this is called endark condition, very characteristic feature of the stem of either it is a angiosperm or a gymnosperm. So here that is a normal feature, and the secondary growth also happens in a normal way. But there are few, at the angles of the stem we observe in the cortex region vascular bundles where the xylem strands are inverted, like that is a different kind of arrangement is seen, and the secondary growth also occurs in these 4 vascular bundles in a limited manner. So it's like unusual presence of the vascular bundles in the cortex region makes it is very unique in the anatomical features Nyctanthus. And this is strychnos, where anomalous secondary growth happens, as you can see here. So the normal way means the whole thing need to be secondary growth, secondary xylem. When the secondary growth happens, 2/3 of the newly formed tissue will be xylem, and only 1/3 or even less than that will be the phloem. That's the usual feature. But here, what actually happens is the xylem, in the case of other Bignonia, we have seen the phloem formation at right angles, at cross like arrangement of the phloem inside the phloem. Just that is what I call as I told it as intruded phloem inside the xylem as it has been. But here, here and there, instead of developing the xylem elements, the cambial ring is going to produce phloem. So the phloem here and there is going to form. So it is called as intraxylary phloem. And also the phloem islands like, the phloem is islands like in the secondary xylem, and it is intraxylary, in between intraxylary xylem, that is very unique to the strychnos. So this is one of the medicinal plant also. Some of you must be aware of Strychnia nux vomica, a poisonous plant and also an important medicinal plant. And this is most of you must have seen nowadays, because of the COVID time, many people are shifted to the hobby of gardening, and many people are growing several plants. So this is Dracaena. So there are several species of Dracaena. We may not be able to make out also exactly what Dracaena is, but this is common Dracaena. We call it pencil Dracaena. We grow in pots as well. Okay, so the interesting point, this Dracaena is a monocot member. Even in the class, I told, like this is a monocot members, as the vascular bundle of the monocot members are devoid of cambium. There is no secondary growth at all in monocots. That is a rule. But here in certain monocots, particularly the Liliaceae members earlier, now they are separated as Agabaceae and Yucca. In all there are several genera very close to one another, like Yucca, Dracaena, Aloe vera. In all these members, we observe secondary growth. So as a rule, there is no secondary growth at all in any monocot member. But when it is happening in some plants, means definitely it will be some abnormality. So anomalous secondary growth is seen in certain perennial monocot members, more commonly in the Liliaceae. So this is the example which we are going to study as Dracaena. Again, this is very important material for your theory as well as practicals. If you practice the diagram very well, both Boerhaave and Dracaena, either of the one you are going to get if they are going to ask about the anomalous secondary growth. That's why I just want to show. So this is how the um, vascular bundle arrangement in the case of Dracaena has been seen. Okay, so
Afternoon, ma'am.
the
Excuse me, page
yeah,
number, please.
please, any doubt?
Page number, please. Good afternoon, ma'am.
Oh, this is in the anomalous secondary growth, anomalous secondary growth of this is Dracaena. You can check once. I didn't hold the study book with me right now as I am showing the slides. It is in
Okay, thank
the secondary growth. Yeah, you will be able to find out.
Okay, ma'am.
Okay,
Thank
so
you.
the anomalous secondary growth happens in the Dracaena is that the vascular bundles are scattered. That form of the basic feature is seen. Another feature of the monocot vascular bundle, stem vascular bundles are concerned, they will have a sclerenchymatous sheath around the vascular bundle, which is called bundle sheath. So here the outer double row of cells, mean thick wall cells, as a double line will show, to show that they are thick wall cells. Okay, so the outer ring is the bundle sheath and inner to that is the xylem. Okay, so that is one of the feature common to monocot members, monocot stem. But here in Dracaena, the vascular bundles are going to form from the cortical region as well, and they show the secondary growth. The parenchymatous tissue is going to transform into the meristematic region, and it develops the vascular bundles. So the newly formed vascular bundles, which are derived from the cortical region, outer region, there is no cortex also in the monocot stem. So they are going to produce the phloem inside and xylem outside. That's why they are called as extraxillary vascular bundles, amphiploic vascular bundles, like they are. So that is one unique feature of the Dracaena. This is how actually it looks when we do the section cutting. I think I have shown you this in the offline class also. This is how it looks like, because the sclerenchymatous tissue as well as the xylem both are going to take more amount of safranin when we stain the slide. Okay, so very clearly it will be shown. So the bundle sheath followed by the xylem elements and the phloem inside. So that is the intraxillary. Sorry, phloem. The phloem is inside, and the xylem is outside. So that is a very unique feature we observe in the case of Dracaena. Okay, so this is the ground view. This is how the vascular bundles are seen in the beginning. But slowly, when the secondary growth happens, compact arrangement of the basilar bundles is going to form, and they are very special in their anatomy. Yeah, so this plant you must have seen. So this is also Dracena. That's what I told you. This is generally the nursery people will call it as Song of India. I don't know the reason, but they call it as. So this is also Dracena, and this is one different tree form like Dracena. Dragon Dracena it is called, and it yields something like a blood-like sap. So that's why they call it as a dragon Dracena. It's more commonly found in the China region and also in the arid regions of Arizona also. So this is beetroot section kettle like that. That is also in the roots also we observe normal secondary growth like in all the dicot members. But the beetroot being a tuberous root, normally in the tuberous roots we don't observe any kind of secondary growth. But the beetroot shows a typical kind of secondary growth, abnormal secondary growth in fact. So here also concentric rings are going to form, but the cambial ring is going to produce like secondary xylem and phloem as sometime, and medulla, the parenchyma tissue for some while. So patches of secondary tissues are going to be produced. So if you observe closely, I hope I have made that also. Yeah, this is how we observe, no? Like the colors wise and also the layers wise, we are able to observe in the case of beetroot. So that is also one interesting kind of secondary growth has been seen that tuberous roots of beetroot. Just out of interest, I just uploaded some. I mean, want to show some of the photos of the plants which we discussed in the offline class and online class as well. This is Magnolia, Michaelia champaka. I mentioned one family in our class, one of the primitive most family called Magnoliaceae, very close to NNAC. I hope at least few of you must have remembered this point. Magnoliaceae family and a Winteriaceae family are the most primitive families among the Angiosperms. So this is one of the magnolias in number gardens. Just that's I would like to show like so that you can identify and remember something about magnolias. Okay, these are some wood elements and all. I think I have tried to show like how the xylem elements and all. So this is Bombax tree, and the wood elements, I think you must have given some assignment work like so. The wood elements will be having in the secondary wood also. Some of the vessels will be very large. So in the cross section, they will be seen like big circles like. So based on the nature of this presence of this big vascular vessels, how they are distributed makes the character of the wood. So this is diffused porous wood we call, because the large vessels are not confined to one region like a one row or just to one region. They are scattered, right? So as you are able to see, they are scattered. So this is diffused porous wood, and more strong in fact. This is how also the tangential section, the how this will be. So this is the longitudinal radial section where we can see the medullary rays, how they are arranged. So this is a surface view, how the scattered large vessels are being seen when we take a section of the wood. Now we Are you able to see the screen? Can you hear me?
Yes, madam.
Yeah, are you able to see the screen?
Yes, madam.
Yeah, thank you. I told a few things I'd like to just discuss with you. Like these are certain things which are not traditionally been discussed in the taxonomy, right? So taxonomy means we always think that is the morphology which is going to help us to identify the plants, and much easier also. That is very important. Of course, for practical purpose, 95% we rely on the morphology only, but there are certain features has been discussed in our syllabus. Like there are recent trends in plant taxonomy. One is about the anatomy. So we are going to take the example of anatomy in the information about the anatomy, particularly to solve certain problems of the taxonomic concern. So taking the help of anatomy, just now we mentioned like the dicot stem will be showing this kind of vascular bundle arrangement, and monocot members will have this kind. So that is a broad level of much more than that. So here the Metcalfe and Chalk Bailey in the year 1954 and 51, and even Metcalfe and Chalk in 50, they have studied so much of information about the epidermis. So that is the, as we know, the epidermis is made of parenchyma. And also there is some specialized cells in the epidermis. They are called as stomatal cells. So that is guard cells surrounded by some specialized cells, more specifically around them. They are called subsidiary cells. The whole thing we call it as a stomatal complex. So part of the epidermis is a stomatal complex, as well as some growths from the epidermis which are called as trichomes. So there are so much specific features about these stomata, their development, and also the trichomes and all put together is going to, this information helps us a lot in the study of taxonomy, particularly to establish the phylogenetic relationship. So here a few things as I'd like to discuss with you. So here, as the epidermal characters wise, stomata is the first thing. So stays in 65 and Vasic in 89. So they just identified some of four types of stomata. So these are the four types of stomata. First, they called as ranunculus type. These are the names given earlier, as I told you. So this is in the case of the terminology wise, here Stace and Vask has given these names. as they are identified this particular type of stomata in these families. As you can see the names here, Ranunculaceae families is one of the dicot family, primitive family as well. So it has been studied in that family. So this ranunculus type of stomata later on been called as anemocytic stomata. The terminology here, the names here, these are the recent names. Recent means the Metcalf and Chuck in 1950 has given these names, but earlier they were called with these names. As you can see the examples of the families here very clearly, as they are studied in Ranunculaceae first, that's why they call them as Ranunculus type. But based on the arrangement of the subsidiary cells and the gods, mainly the subsidiary cells number and their arrangement wise, they are named differently. These are the names presently we are using. Okay, so just to see that ranunculus type are the anemocytic here. It is
Excuse
anemocytic
me,
means
ma'am.
the
Screen is not
subsidiary cells
visible.
are not
Excuse me,
specific.
ma'am.
They are just like the
Screen is
normal
not
epidermal
visible.
cells. That's why they are called as anemocytic. So here it is
Excuse me, ma'am. Screen
example
is
of
not
this family are
visible.
Ranunculaceae and Malvaceae. Then anisocytic.
Ma'am, excuse me, ma'am. There is no screenshot, ma'am. There is no any screenshot invisible, ma'am.
Are you not able to see the screen?
No, ma'am, no.
Oh, sorry. Just just 1 minute, 1 minute. వాళ్ళ స్క్రీన్ కనిపించలేదు ఆడియో. I hope you are able to see the screen now.
Yes, ma'am. Visible, ma'am.
Thank you, because I thought you are able to see and I am discussing. Okay, so this is what I'd like to show you, like how the anatomy is going to help us in the study of taxonomy. Yeah, you heard, no, whatever I told. So these are the things like the type of stomata, as we know, like there are anemocytic, anisocytic, parasitic, diacytic stomata. Particularly these four types of stomata are being referred or identified in the beginning in dicot members, and they are very common also in the dicot members. So the anemocytic means the subsidiary cells which are around the guard cells are called subsidiary cells. They are not significant. They are just like any other epidermal cell. That's why they are called anemocytic. They are seen in the families like Ranunculaceae and Malvaceae. Then anisocytic means they are not similar. That's the meaning, aniso. Like we use this term in other instances as well. Anisogamy, something like that, right? So anisocytic here, this is seen in the cruciferous families mainly, that is Brassicaceae right now, but also it is seen in the Solanaceae members like the Datura and all. So here there will be three subsidiary cells. One will be much smaller and two will be larger. So as they are unequal in size, it is called anisocytic. So significant. We are picking up only two families here. So it doesn't mean that they are present only in these families, but often seen more number of the stomata in these families. That is one point you need to remember. When we are just mentioning running some family names here with this particular kind of stomata, it doesn't mean that that family members shows only that type of stomata, and it is not seen in other members. That is one thing. And any plant or any leaf will have at least minimum of two different types of stomata or even more, but never only one type of stomata are seen in any plant. So that much you need to have the clarity. But as we are identifying these with certain families means more number of that kind of stomata has been seen in these families. I hope you got the point. Yes, next is the
Means in one leaf we can see more than
parasitic
one type of stomata, ma'am.
parasitic and ascitic stomata where the subsidiary cells are only two. So they arrange stomata where it is also called as rubiaceous type.
Excuse me, ma'am.
So
In one
here
leaf
and
we
also
can
parallel.
excuse me,
The two
ma'am. Excuse me, ma'am. In one leaf we can see number of this type of stomata. Single leaf have
in one leaf
single leaf will have more than one type of stomata, ma'am.
at least two types of stomata are seen. At
In
least
single
two
leaf.
types. Hibiscus leaf. Here I told it is the Malvaceae leaf. Yeah, just try to follow me what I'm saying. So here I have given example of anomocytic stomata in Malvaceae. So I can take one hibiscus leaf. When I taken one hibiscus leaf means it will have more number of anomocytic stomata, but they'll also will have anisocytic, right? But few in number. Similarly, if I told like the parasitic
According to
stomata,
measure.
yeah,
Excuse me, ma'am. According
more
to majority of more
number of stomata are of
number,
this
we
type.
can name them as a
Yes,
anomocyte.
more number of
Large number of
stomata. That's why we are giving this as an example. So that's what yes, more number of stomata in that leaf will be of anomocytic
for
in
example, hibiscus has in single 80 anomocytic and 20 anisocytic. It mentioned as anomocytic.
Analytic, I have given the example of Solanacino.
Ah,
So when we are observing Datura, there will be three types: anomocytic, anisocytic, parasitic. Three types are same.
means according to majority of
But
stomata, we can
more
name.
depends in certain. Yes, we are just taking that as an example. But these are actually the type of stomata. Okay, so here the parasitic stomata is the one where the two subsidiary cells are parallel to the gut cells. Parallelly, they'll be arranged. So that is seen in the case of Rubiaceae and also Magnoliaceae members. In the
Rubiaceae means
diacytic, here
sapota.
also the
Rubiaceae means sapota.
no. Rubiaceae means Ixora.
Jasmine.
Ixora.
Okay.
Ramavana antarade Ixora flowers, beautiful bunches of red, orange, yellow, white, many fragrant flowers are seen in Rubiaceae.
Okay.
And a coffee plant belongs to Rubiaceae also.
Okay, okay.
Yeah. Okay. So this is diacytic is the fourth one. Here the two subsidiary cells will be diagonal at right angles. So like this. Earlier I told parasitic parallel to the gut cells, but here at the diagonal they are present. That is the diacytic stomata. So this you can clearly see here Magnoliaceae I given in both the cases parasitic diacytic. Then you I think now you will be able to pick up, right? So in these two types are seen in Magnoliaceae, not only one type. So this is how actually the some plants with the more number of a particular type of stomata will be able to identify based on this type of stomata as well. So these are the four types we usually observe in the dicot members, and similarly there are different kinds of suggested in the monocots. More variation is seen in the monocot members regarding the stomata are concerned. So mainly they are also called as tetracyclic, means two diagonal and two parallel are seen, that is tetracyclic stomata are seen in monocots, and actinocytic here four radially elongated subsidiary cells, all four are at both all the sides it is seen, it is called actinocytic stomata. And cyclocytic means four or more cell, number of cells are being more, four or
Stomata.
even much more, and they are around the guard cells like a ring-like manner, that is cyclocytic. And hexacytic means six subsidiary cells, four parallel and one two above and below, okay, at right angles. So these are different types of stomatal types, as we are clearly able to indicate these are present in certain families. So that means in a way that it is an identification character, is it not? Similarly, in monocots, we are finding along with these types and the additional types are also seen, which are definitely will be useful to the family level, not genera species level. And also another feature regarding the subsidiary cells only, I hope you are able to see. So how the stomata has been distributed is also an important thing, like epistomatic when they are present only on the upper epidermis, hypostomatic they will be present only on the lower epidermis, or amphistomatic, so on both the sides. So we all of us know mostly in the xerophytic plants, the epistomatic condition, the stomata will be seen on the hypostomatic, they are seen on the lower surface to reduce the transpiration, right? So similarly, epistomatic is the character of the hydrophytes, where to avoid the contact of water in the lower surface, they are much less and they will be more on the upper surface. So in the mesophytic plants, on both the surfaces they are distributed. So the distribution is also is very much useful in certain times. So here the epistomatic stomata is characteristic, like here I take the example of ulnus subcoordinata, and they are hypostomatic in ulnus orientalis. So here, so in different species, the stomata are present in a different way. So sometimes the presence of the stomata on which surface is also helpful in identification. So here clearly in the ulnus species, we are able to use this character epistomatic and hypostomatic condition to distinguish the two different species. In ulnus subcoordinated it is epistomatic, and here in the orientalis it is hypostomatic. So here I told it is useful to the types wise, it is useful to the identification of the families, but the distribution wise sometimes useful to the level of species also. Which remember one thing, whichever is the character useful to the species identification is more significant in systematics. That means with that particular character, whatever it is, not only the anatomical features, if it is useful for the identification of a species, means it has lot of significance in the systematics. Just remember that, that is one point. So here this is perigenous, mesogenous, this is based on the shape of the guard cells also differ, all of you know that, right? Normally in the dicot members we call it as bean shaped guard cells, whereas in the case of monocots they are dumbbell shaped. They are very unique. The dumbbell shaped stomata are very unique, guard cells are very unique, their thickenings are also very unique. They help in the identification. The thickening patterns are very helpful in the identification of certain genera in monocots. Okay, then this is the perigenous, mesogenous, mesoperigenous types. These are all based on the development. Like as we know, the guard cells and the subsidiary cells might have derived from one cell, one mother cell, or they might have derived from two mother cells or from different cells also. So based on that, it's perigenous means it is from the same cell. Mesogenous means from guard cells from one mother cell, and subsidiary cells from another mother cell. And if it is in between, guard cells and few subsidiary cells from one cell and subsidiary cells from another cell, it is called mesoperigenous. So mix of both the types. So this is actually greatly studied, extensively studied in different the members of Portuguese and IGAC by our Usmania professors Ramaya and Rajgopal. So they are very eminent anatomists and also taxonomists, and leading. They were the leading epidermologists. Epidermology studies wise, they actually the Rajgopal sir used to say like even a small plant material from the surrounding Hyderabad, he could identify that much of extensive work has been done. Not only these two families, almost all the flora of Hyderabad has been studied. Rajgopal sir and Ramaya sir, a great contribution to the taxonomy and also anatomy from the Usman University. Then another thing is about the trichomes. As this is about as in the part of the epidermis, anatomical studies means not the pure anatomy. I told it is based on the epidermal part only, epidermology. So here the trichomes are going to be very much helpful. So here the Theobald et al. In the 1979 used the classification of the trichomes like here the extensive study as already just now I mentioned about Ramaya, Professor Ramaya. So Ramaya sir, he has given a key to the identification of the composite members based on the trichomes, and Stace has given about the Combretaceae family. So they are very much helpful in the identification. The trichomes are the epidermal growths. There are very different types of trichomes are being seen: unicellular, multicellular. More commonly, at least as we know, Malvaceae as a known family to all of us. So the Malvaceae members are very unique in having a special kind of trichomes. They are called stellate hairs, star shape. Normally, just a growth like simple unicellular structures are being seen, but star-shaped trichomes are the important character of the Malvaceae. Similarly, glandular trichomes are very characteristic feature of the family Lamiaceae. Ocimum family is known for having glandular hairs, and even Solanaceae members like the Datura and all are having very unique trichomes. They are glandular trichomes and uniseriate trichomes and unicellular trichomes like that. The trichomes are going to help a lot for the identification of several species, genera and families. Okay, so these are epidermal cells like. So the you have a very detailed information in the book. Just go through once, not for the exam sake, just for the idea how much of information we can take from anatomy, particularly with the epidermis, to identify certain plants or the diverse device, how it is helpful. So the conquest and use this epidermal cell morphology as well. So Cranquist, so he studied the angiosperm leaf diversity so much, and also IMC in the 1974, the sclerenchyma patterns in Willowiaceae he has been studied, and Govindarajulu, he is from Kakatiya University in 1969. He has studied the epidermal cells patterns in the case of Cyperaceae members for the identification of genera. So the epidermal cells means the shape of the epidermal cells. They might be like angular, the contour as we call the outlines of the epidermal cells might be straight lines or crooked lines or with so much of convoluted margins, and also they are very helpful in the identification of certain several families, and these are some specific contributions from the epidermal cells only for the systematics are concerned. And normally we think anatomy means we'll study about the structure of the root, structure of the stem and leaf. Is it not? That's what generally we think. But in the plant, there are, particularly in the case of dicots, and also, of course, even in the gymnosperms as well, there will be a petiole, the shoot. We discussed, no, the shoot apex will be having lot of complexities and all. So here there is at the petiole, as the petiole is something like the stalk of the leaf. It arises from the stem, is it not? So the vascular bundle, so how it is going to enter the petiole, that is very interesting to study. The petiole anatomy and the nodal anatomy, just go through once, just to have an idea how the diversity of the forms are being seen. So there are called the leaf traces and leaf gaps are been there. So in the petiole anatomy, that also helps in identifying whether they are advanced type or primitive type, and what are the different links between the primitive and advanced type. Based on the petiole anatomy also you can identify. So as just some of the words like unilacunar means only one space will be there at the petiole, or trilacunar or multilacunar. So several gaps will be there in the vascular supply to the petiole from the stem. So based on that, unilacunar, which is considered to be more advanced, and trilacunar and multilacunar are different forms are seen. So by observing these things also we can think about whether they are primitive or advanced. Okay, similarly, wood anatomy. So as we know, the wood anatomy means just in the last few slides I have shown you what the type of vessels are being seen, how they are arranged, and what are the elements of the wood. Like sometimes in the gymnosperms, we know the gymnosperm wood is not heavy wood. Heavy wood means not a durable wood because they have only the tracheids, not the vessels. Even though it's pycnoxylic and compact cells, the thickening is not that much of the vessels. So the dicot members yields a better quality wood than the gymnosperms. So that wood anatomy. So this is one thing I told you. The vessels are considered to be more advanced as they are present in all the angiosperms except few. Right? This is the family I mentioned, Vintraceae. Right, so the Vintraceae family is one of the dicot family
Yes,
without vessels.
vesselless.
Yeah, vesselless family. That's what I told. So vesselless family wise, it is. That's why I told you it is the primitive most, and Magnoliaceae members are primitive members. Okay, just to say that why that the primitive most, as to say, because vessels are more advanced and they are seen in dicot members. So this is one family where it is absent, and there are very trichodentraceae and other members which are very close to that. They are having different kinds of vessels. Which are of, again, in the vessels also certain features has been seen as primitive and advanced. So that whole thing we can just take it for the establishing certain relationship among the primitive families and advanced families. So that means wood and wood anatomy also helps, and similarly the floral anatomy. Eames and Puri, Murthy and Puri. So these are few member scientists who worked on the floral anatomy. Means flower is much more complex structure, is it not? So the flower, as I told you, is considered to be a modified shoot apex. So instead of developing the leaves and leaves as a shoot apex, it developed as a flower. So naturally, each part of the flower, the calyx, corolla, androecium, gynoecium will have the vascular supply. So how it is? So that is very interesting that floral anatomy will be more complex. So it helps more distinctly to understand the floral morphology in Papaver, Capparidae and all. So these are the scientists actually studied the um, different features of this floral anatomy of these families, which helps in understanding whether they are advanced or primitive. They established some, based on the floral anatomy, they could establish the position of these families. That's one of the um, reference we can make. Then this is very recent, in fact, fruit wall and seed coat also been used in taxonomy. So particularly the seed coat anatomy. This is the seed coat structure that also been used in the classification. It is actually called as Corners classification. Like along with other features, they also consider the seed coat how it is in. So here it is Amania and Rotala. So they belong to the family Lythraceae. So other features are completely different. So based on the seed coat anatomy, they consider these two are related, and they retained in the family called Lythraceae. The Lythraceae family is our henna family. And similarly, there is one genera called Legasca, very very common weed plant with white small globose flowers like Astraceae member Legasca. So it is been placed in Helianthe. That is, I hope some of you must be knowing the sunflower name Helianthus. So it's like Helianthus is a heterogamous head, right? Having the disc florets and ray florets like. So that kind of head inflorescence having members are kept in a tribe called Helianthe. Okay, so the Legasca will be having only disc florets, but still it is kept in Helianthe because as they observe the seed coat anatomy is similar. One of our contributing for the textbook, Pullaya sir from Anantapur, Sri Krishna Devara University, he has studied the seed coat anatomy of this Legasca and suggested it is need to be retained in Helianthia of Astraceae, even though it is a homogamous head. So that means the seed coat anatomy also helps in establishing the position of certain genera, whether to retain in the same tribe or family or even their relationships, as well as I told you here, these two, because of this similarity of the seed coat anatomy, retained in the family Lythraceae. Okay, so this is a recent thing, center like ultra structure of the relation of the taxonomy. So there is like the sieve proteins, and this is what actually the latest thing, sieve proteins and P type and S type of proteins have been seen in the sieve tubes. So based on that also they could identify that is more specific, not just only the anatomical features, it is more ultra structure wise it has been seen. Okay, so this is about use of anatomy. I'm just concluding with this. So in brief, I would like to add some of the things like just at least so that you will feel familiar with the topics which are there in our syllabus. I hope my voice is clear. Am I audible?
Yes, ma'am.
Yes, yes.
Some notification like the audio system is weak, something. Okay, so quickly we can look into the cytology part. So I hope you know the what is cytology. So here it is the chromosomal study. The cells will have the nucleus, and there is the chromatin reticulum, and it is going to make form into a chromosome. So that whole thing, you know. So the use of that chromosomes in the study of the taxonomy, that is a role of cytology in taxonomy. That's what actually the, so as all of us know, the chromosome number is constant to every species, almost 100%. Just a slight variation might be, but in general, it is the most standard feature we observe in all the life forms, not only in plants, in animal, that is a life forms. That's why I'm using this word. In all the life forms, chromosome number is one of the constant feature without any exception. So there will be starting from the least amount of the haploid number, as we call the haploid number, from 2 to the highest number can be up to more than 600. So there are mostly the members have been either the lower groups of plants only exist in the form of haploid condition, but other than from bryophyte onwards, it is only the diploid plants. But when it is proceeding to the higher plants, the diploid condition might not be seen in certain members like maize or most of our cultivated plants now, either it is cereals or pulses or the anything, any type of plant, they're all polyploids. So in wild, we might be observed some diploids. So that is the evolution point. So from haploid to the diploids and polyploids are being seen. So based on the chromosome number, as I told, it is constant. But the chromosome morphology is one of the important feature. So we can just arrange the chromosomes according to the type and their size. So that arrangement of the chromosomes according to their size and the types is called karyotype. It's a karyotype analysis we call. So that helps a lot because the chromosome structure based on the centromere position and all, as we know those terms and all. So and even the chromosomes might be having primary satellites, secondary satellites and all, and sometimes even some B chromosomes and so that helps a lot in identification of several species, right? So here. Then, of course, just now I told you there will be many polyploids also, or some aneuploids as well. So, the number of that chromosomes helps in understanding that karyotype information, and more than that, it is the meiotic behavior of the chromosomes. So that is more specific, not all the time, but karyotype analysis helps us more. And meiotic behavior of the chromosomes means if they are as the chromosome number, based on the chromosome number, more often it is the even number will be seen. What are the polyploids ultimately? Because we know that in the case of meiotic division, half the number of the chromosomes will separate. As they are homologous chromosomes, they need to get separated, right? So the paternal and maternal set. So if it is some uneven number is there, it will not be able to distribute equally to both the sides, right? So that kind of some kind of meiotic behavior also shows some kind of how special they are. Any type of lagging chromosomes are there and any uneven distribution happens like that, so that gives a kind of clue how they are related, or any of special features has been seen in that particular meiotic behavior also helps in understanding the specific nature of certain genera, not at the species level. So the cytology is going to help very much in solving several problems, particularly with the help of the karyotype analysis. And the next thing, as I told you, chemotaxonomy is another interesting thing, and this is up to from 1919 onwards, like 90s onwards, so up to 20, 2000 or even 10 years back also, the chemotaxonomy is one of the important information the taxonomy is used to take to establish the phylogeny relationships more often. Because they are very specific, the chemical substances, as just now we told about the chromosome structure. So the chromosomes are ultimately going to be the source of, like that is a genotype, so they are going to be reflected in the form of a phenotype. So as we know, the chromosomes, the genetic material in fact is going to make the difference of their chemical constants, chemical substances present in each plant may not be the same, or some plants will be having certain kind of specific substances. So here a few things, like just to understand how important it is. The plant material will be having the basic structure is composed of carbon, hydrogen, oxygen, nitrogen, right? So those are the main components. Even they make the plant body, the cell wall and cell organelles, everything. So as the metabolic process starts, as it functions, so the the substances which are involved in the normal metabolic process, they are called as primary metabolites, like the carbohydrates or the amino acids, whatever the general things which are related to our photosynthesis, respiration, and protein synthesis. All these are all will be called as primary metabolites. They will be common for all the plants. At least for all the plants, they are common. Autotrophic plants, at least, leaving the fungal members aside. So they are common. But there are certain substances. They are generated in the process of metabolism. They get accumulated. They are never recycled, and they are very specific. So those are called as secondary metabolites, right? So these secondary metabolites are very specific to certain plants. So here, remember, the secondary metabolites are very unique to certain families, certain genera, certain species. So that's why they are very important for the classification, like from the crankwaist onwards. Crankwaist, thorn, there are different classifications has been made in the angiosperms. So they're all taken so much of importance. They are given so much of importance to this chemotaxon because it appears more credible than based on morphological characters. If you are just trying to relate the plant, relate the species, or relate the genera belongs to a family, more than that, this this appears more scientific, is it not? As the same type of chemical substance are found means it is truly related. Like that, the secondary metabolites study has been gained so much of importance, and that develops as a science called chemotaxonomy. So here the secondary metabolites can be of different kinds. They can be alkaloids, steroids, or tannins, resins, or glycosides, or even sometimes to the level of some amino acids also can be different. Not very much, but sometimes. Okay, so the classification here we are talking about classification of rhodial families, like cappareals like Brassicaceae, Capparidaceae, Rosidaceae, and Moringaceae. These are all belong to the order Cappareals, and the papaverils were with the papaverase and fumariaceae. So based on the chemotaxonomy, they found they are related. And similarly, classification of the order Centrospermae. Here they are taken the pigments as an evidence, like the betalains, flavonoids, and all those things are anthocyanin pigments. They are taken as evidence for the classification of Centrospermae. And here that's what I'm saying, like leucoanthocyanins are found in primulaceae. They are absent in woody members of the Oleaceae and Scrophulariaceae. So by observing this leucocyanthanins in the primulaceae, they are found, which are not to be seen in Oleaceae and Scrophulariaceae. So here we are taking only the evidence of ultimately the last aspect we are considering is about the chemotaxon. It helps a lot, and even it is also called as, by adding the chemotaxonomy in the systematics, like the Cronquist or Tuctigen classification. It is also called as omega classification or synthetic classification. Okay, so another point, like just a word, like there is one word called cementioids. The macromolecules found in the plants, they are called as cementioids. Like as just I made a word here, I just mentioned like DNA and RNA. So the cementioids are the macromolecules found in the plants. So there are identified as three types of cementioids: primary, secondary, and tertiary cementioids. So as I told, macromolecules like the DNA and RNA and proteins, these are all macromolecules having more molecular weight. So the DNA being the genetic material, and more importantly in all the life forms, being the genetic material and much more having molecular weight, that is called the primary cementioid. And the RNA is the secondary cementioid, might be hereditary in viral particles and all, but as the different types of RNA are comes under the category of secondary cementioids, which are very specific. So naturally, we know the genetic as presently the genomics and all the DNA sequencing, everything is when it is happening. So definitely it matters a lot. The primary cementioids, secondary cementioids, and the last one is the proteins. So as the proteins derived from the genetic resource from the DNA and RNA at the end, the third level, the proteins also differ. As they are macromolecule, they are also called as non-genetic macromolecules, while the proteins are called as tertiary cementioids. So all these three are having lot of significance in their own way, their function wise, and also they are useful in the systematics as well. That's what presently the genomics or proteomics. The latest things are proteomics. We are already left the genomics as well. So the proteomics, ultimately they are the DNA is going to make the RNA, and RNA is going to provide the information as to make the proteins, right? So that will be the ultimate. The end product is the protein. So the protein changes, how they are been there, is also studied, and they're also used in systematics as well. Okay, so that is the latest developments wise, not only in taxonomy, they are the latest technology, proteomics. So this is one thing I told you about the numerical taxonomy. This is there. So this is another thing. Of course, not easy to explain. Like, so the numerical taxonomy, I just briefed in our class also. Like the computer is there in all the fields. So here we are going to use the computer knowledge in taxonomy, which will be like another expression. We are trying to explain taxonomy in different ways, right? Or just now we told like the recent trends. So how the taxonomy is taking the inputs from other fields of science, but here we are using the computer, like the taxonomy is expressed in terms of numericals with the number. So far we have just based on the phonetics, the visible characters or the chemical characters wise we are trying to explain the taxonomy, but here with the number. So that is with the that is what actually the numerical taxonomy means. So here it is the Haywood has been begun with in 1967, and Michael Anderson has given the protocol how to use the number. Means here, as I told, it is the computer data wise we are going to give the characters, and based on the weightage of the characters, as they are called as Adamsonian principles, they are going to analyze just only as we feed the characters not to the system. So here we are going to give the characters with some points like plus and minus. So 2 digits need to be entered in the computer. So it need to be either it is the characters will be taken. They are given as a plus and minus like for every character. So accordingly they feed the characters to the system, and finally they consider so how many characters are matching and how many characters are not matching. So here this is the numerical taxonomy wise, it is empirical science and aims to develop objective method. So here there the main term we need to remember is operational taxonomy groups, OTU groups. That is what we are going to make by using the characters with some weightage, like assessing them, and that is the OTUs are the basic unit. It can be individual species, genus, family, order, or class. That means the numerical taxonomic data we can apply to any level. Okay, just for your understanding, I try to give a few things about numerical taxonomy. Just go through once how it is. And it is something like ultimately after analyzing the characters we are going to make the cluster analysis. Means just data will be given, and we'll be able to give how many OTU values are been there, and by combining them we can make a cluster. Okay, so from where which group is the beginning, and how it has been divided, and how it has been multiplied like that. So that is called as a cluster analysis. The different OTUs grouped on the basis of similarity. So this is they call it as phenons and ranks. Groups are obtained from the cluster. Will help us to understand the phylogeny. So the numerical taxonomy has a lot of significance in making the phylogenetic relationships, because by using this analysis we'll be able to make the groups. So very easy to what is the first source and how it has been divided and how the other things are. So we'll set up the things first, the different groups, that is the operational units we are going to pick up, and we are going to pick some common features, and we can make the next level of group, and ultimately the major groups we are going to make something like a dendrogram reverse way. We are going to get the results here, which helps to establish phylogenetic relationships. That is the application of numerical taxonomy. Okay, so this is where I'd like to end the session. Most of the things I completed in offline, and also I try to give few things which we have missed in offline. So that those things I covered in the online class now. So today I am ending the session. Whichever is the doubts you are having regarding the subject, you can just send a message. I can clarify. Or at any time you can call me also after 5 or 6 you can call me if you have any doubt, and yeah, by evening or tomorrow I'll send you the important questions. I'll a PDF I'll make, and I'll send the entire list of short questions and essay questions in the course 3. Okay, thank you everyone.
Okay, ma'am. Thank you, ma'am.
You're well.
Thank you, ma'am.
I'll send you to, yeah, positively by tomorrow evening I'll send the PDF of question bank.
Class also, can you send me, please?
I'm,
Excuse me, ma'am.
I'm going to conclude today. I'm not going. So tomorrow you'll not be having my class. So today I'm.
Ma'am, excuse me.
Okay. Thank you, everyone.
Excuse
Thank you,
me,
ma'am.
ma'am.
Yeah. Any questions, please? Yeah.
Excuse me, ma'am. Last week topic also you have given. You want to give questions, ma'am?
Pardon?
Last week class also we need some important questions. Last week class.
Not just last week. The entire from gymnosperms to anatomy. From every unit, what are the important questions? Short question and essay question. Prepared a list of it. I'll send the PDF tomorrow.
Thank you, ma'am.
And just check once the old papers. You can ask sir about the old papers. Then you will know the final version of what are usually asked, often asked questions. While discussing the things I just mentioned, what are the things often asked, but still you can see black and white. The if you have old papers, you'll have an idea. Okay. I'll put a star mark, even according to my experience. I'll put a star mark which is the most important. Then you can make a short list of it. Okay.
Okay, ma'am. Thank you very much.
So all the best, everyone.
Thank you, ma'am. Yeah.